acquired mutation in the estrogen receptor encoding gene ESR1 can lead to secondary resistance to endocrine therapy. And our finding is ESR1 gene mutation rate is just 1% in primary breast cancer patients and 18.6% in MBC patients. And the location of the mutated ESR1 gene is similar compared to the Western population.
The mark of my gratitude to the co-chair and also to our staff for their great effort.
Of you, especially for my mentors. Today, my topic is advancing the precision oncology breast cancer, collaborated NGS and studying inside from Guangdong Provincial People's Hospital. So the breast cancer is a global health issue, ranking first in incidence among female cancers. So before 2000, when I studied in Europe, we know that the standing, the pathological outcome and the TNM stage are the basic standard for the treatment. But after 2000, we have a breakthrough that in that year. Several important published paper in natural science and show that we representing a milestone in breast cancer management. Transcriptomic profiling has stratified the disease into 5 distinct molecular subtypes. The NGS next generation sequencing emerged in the early 21st century, representing a revolutionary leap in biotechnology. And from 1990 to 2010, we have 4 breakthroughs in the history of sequencing technology. So in this background, our question is, in breast cancer, what is the hold of the NGS? And what insight can be learned from this analyzing? And also, particularly in the context of the primary breast cancer, what can we learn that from the cancer tissue? So actually, dissecting the genomic landscape of breast cancer, and we have made huge progress in this domain. And NGS enables the comprehensive mapping of the genomic landscape in breast cancer. As we know that from the first paper from the 2012 from the Nature TCGA book, published important paper, the first time in breast cancer, comprehensive molecular portrait of human breast tumors, and with 825 breast cancer samples. And for our team, I work with my students. We work focus on NGS too. From 2007 to 2019, our team conducted in-depth NGS research and mapped the molecular landscape of breast cancer. And also, our finding published in 2019. Our finding in this paper, we could see that compared the Western population, the most frequently mutated gene is TP53 gene, and it's 45%. And followed by the PI3K gene mutate, this is somatic mutate, it's 44%. It's higher than the Western population. And then the third one is HER2 amplification or HER2 mutate. It's 24%. and is higher than the Western population too. And go back to the genetic gene mutation. As we know that since 1994, the BRCA1 we understand well, and by the Professor Merker from the United States. And approximately the 5% to 10% of breast cancer are driven by Gemini the past or GE variant. And from this paper, JAMA, and published 2017, we could see that at BRCA1 and 2 mutation are the most common, with carrier facing a lifetime risk more than 70%. And our team established the mutation spectrum of the hereditary related gene in Chinese breast cancer patients. And our findings published in 2020 February in journal Aging. And our finding in this research, we find that in Chinese population breast cancer patients, the most frequently mutated gene is BRCA2, 3.44%, but not BRCA1. And most of these patients carry with BRCA2 mutate. Their subtype always is HR positive group. And the second Gemini gene mutate is BRCA1, is 2.10%. And the fourth, the Gemini gene mutate is EALB2, is 0.76%. And then follow is CDH1 Gemini gene mutation, and it's 0.57%. And ID51C is 0.57%. This is our finding, and so we could find that there is difference between the Chinese population and the Western population in the Gemini gene mutation. And furthermore, this year, we just published another paper in the Discovered Oncology. And the pattern of gene, the Gemini, the variant in breast cancer varies significantly by HER2 expression status, especially HER2 low and HER2 zero subtype are enriched for BRCA and SPANC pathway variant, where the HER2 higher type mirrors the general population. And in this paper, we had important information. And also the Professor Jennifer gave us many help. So we work together for this paper. So we are so happy that we could share that this important genetic gene mutation in this big panel 102 gene, included 102 gene, the panel for the genetic gene mutation. Then we come back to the somatic mutation and go back to the ESR1, the mutation. From the published paper 2015, ESR1 mutation data detected in metastatic ER positive breast cancer with 187 cases. In this paper, the finding is the most common mutation type included 537 or 538. So acquired mutation in the estrogen receptor encoding ESR1 can lead to secondary resistance to endocrine therapy. So after this outcome, our team focused on ESR1 research too. We published our outcome, the data in 2020 in Oncotarget. And ESR1 mutate status in Chinese breast cancer in Guangdong Provincial People's Hospital with 270 cases. And our finding is ESR1 gene mutation rate is just 1% in primary breast cancer patients and 18.6% in MBC patients. And the location of the mutate ESR1 gene is similar compared to the Western population. Now we go back to the HER2 receptor signaling network. And I think we understand this pathway very well. And now for the HER2 breast cancer and anti-HER2 therapy, actually we have many regimens, and we also have the standard the treatment. And Professor Guangdong just help us update the new data in the HER2 group. And from the DB05, DB09, and DB11 trial, we have the new data. And what is our finding concerning HER2 pathway related research? And this is the first paper and the first research in the HER2 domain. We published in 2020 in breast cancer research treatment. And in this paper, actually we share genetic mutation profile of Chinese HER2 positive breast cancers and genetic predictive of response to neoadjuvant anti-HER2 treatment. And our finding is there were significant differences in baseline Ki67 value, TP53 sense mutation, TP53 loss function mutation, and PI3K mutation, and NOS1 mutation between the PCR group and the non-PCR group. And another paper and another research is published in 2022. In this, we talk about what is the difference of genomic profiles in patients with HER2 positive breast cancer. I mean, group HR positive, HER2 positive, and compare HR negative and HER2 positive. And in this research of finding that the variance of the HER2 family in the HR positive, HER2 positive group and the HR negative, HER2 positive breast cancer group. We could find that in HR positive and HER2 positive, they have more HER2 amplification at the same time with the HER2 mutation. And also in this group, they have more HER3 mutation. That's our finding. And in 2023, in San Antonio, we have another poster, we have another research, and our finding that is HER2 ratio CEP17 and HER2 contribute to screening the patients who may benefit from the neoadjuvant chemotherapy combined with anti-HER2 therapy. Here, we find the two groups, it's a focused amplification and the broad amplification. Focused amplification means just HER2 amplification. And the broad amplification means at the same time, the patient is more another gene mutate. HER2 amplification and red and stage 3 and CDK 12 gene mutate in the same location. That's the different. And this is our the research unpublished data. And you see that, and we focus on the NGS-based HER2 copy number and mutation the profile of the 1,088 patients. And in this research, we could have the finding is focal the amplification group show significantly higher tumor HER2 copy number than the broad amplification group. And also the proportion of the HER2 positive IHC 3+ and FISH positive cases was significantly higher in the focal amplification group compared to the broad amplification group. And in this research, we also find that the copy of the copy number of the amplification of the HER2 and could predict the accepting to the neoadjuvant and the ADC to the neoadjuvant. It means if have the low copy number, it means could be have a good response to the neoadjuvant with the ADC. And go to the PI3K-AKT pathway. In novel 120 trial, we could see that the PI3K inhibitor for adjuvant therapy resistance relapse in PI3K mutant population. And now the PI3K inhibitor is currently the only has demonstrated benefit. And our finding concerning the PI3K-AKT pathway related research is that an activation of the PI3K-AKT pathway promote breast cancer cells and is commonly mutated pathway in HR positive breast cancer patient. And in 2021, we have the finding that in the with the 589 cases, we find that the mutation rate in this pathway is as high as 62.6%, involving PI3K, the gene, 45%, and PTEN is 7.5%, and AKT1 is 5.9% in HR positive and HER2 negative group. And go back to the trial 29-1, and we know that AKT inhibitor work. And our finding concerning PI3K-AKT pathway, and we could find that the research, the Chinese cohort share that distinct AKT mutation spectrum with a significantly higher AKT1 and lower AKT3 mutation rate than TCGA, and driven by the AKT1 E70K hotspot. And this finding published 20 and 21. So with our research and we explore the NGS information in primary cancer tumor, and with our mentor, Professor Ning Liao and Professor Charles Bauch and Professor Susan and many also the Professor Hobart Go and Professor the Sway and Danny and Charles Wyatt and many the experts from the CEN. We organize MTB molecular tumor board is the MDT version 2. And every Wednesday evening, 7:30 p.m., we have an international consult. So we keep this program for 7 years. And in this program, we help many comprehensive patients. We also have international the regimens for our patients. And we could have the chance to discuss how to make a better the regimens and the plan for our patients. So this is one of our case. And in this case, we summary of the molecular result with the germline mutated BRCA2 and also have the somatic mutated P10 and also have the FGFR1 amplification. And also we have the TNN and what is the characterized the patient and the Professor Gradisher and Professor Charles Baus and Susan Klimben and guide us discuss with the Professor Swain and Daniel Hale and Vincent panel. So this help this patient now the PFS continue more than 3 years. So summary from our finding, as the genomic and expression providing data continue to be integrated and optimized with clinical pathological characteristics. Research has achieved significant breakthroughs in understanding the underlying mechanisms of breast cancer. And this will ultimately drive improvement in the biomarker tools, thereby guiding strategies for intensified and better treatment. Thanks your attention.